Can somebody thumbs up on Zoom that they can hear me? Does anybody on Zoom hear us? Yep, okay, great. So let's start. So welcome. This is an Encore Institute event, and we are very excited to be hosting Anqi Wu. Anqi is an assistant professor at the School of Computational Science and Engineering at Georgia Tech. Previous to that, she was a postdoctoral fellow at the Center for Theoretical Neuroscience at Columbia University, and she received her PhD degree in computational and quantitative neuroscience from Princeton University. Anqi is a rising star in EECS, a DARPA riser, and she recently received the Sloan. So congratulations to Anqi. And she's very kindly agreed to give us a 4-hour tutorial for 2 hours today, 10 to 12 Pacific, and tomorrow also 10 to 12 on statistical machine learning models for neural and behavioral data analysis. So without further ado.
Can people hear me well? Online too? Okay. Yeah, thanks, Gail, for the nice introduction, especially the invitation to give this presentation for the Encore Institute event. Really glad to have a chance to travel all the way from the East Coastline to the West and to feel this chill vibe in the West Coastline and also meet people here. So really glad to come. So yeah, before I start, I'm just curious, like, can you just hand up if you don't have, or how many people have some background knowledge in neuroscience in this audience? Okay, pretty good amount, I would assume. So yeah, because yeah, I'm gonna talk about statistical machine learnings. Just how many people have knowledge about machine learning's data analysis tools? Okay, pretty great, yeah. So it's the combination of both, right? You probably need to know something about neural data, but if you don't, I can briefly mention something. It's not too complicated kind of data to understand. And for the statistical machine learning tools, I also try to introduce some basic ones, and hopefully these basic tools can build up to a more specific, more sophisticated tools. Cool. Yeah, so generally the thing I'm gonna talking about is with respect to this neural coding. Okay, so usually we have this brain, which is the black box, very complicated, very hard to understand. We have the input sensory stimuli, like the images, videos, orders, like input to the brain, and generate a bunch of neural signals. We have the spike trains, which are usually these discrete value data from the electrical physiology data, electrical physiology recordings from system neuroscience. People can open the brain of an animal, collect a bunch of neuron spiking events. Or we can have constant imaging, which is more continuous data, still invasive, but you can collect more, a larger population of neural recordings. And then we have also the bulk signal, which is usually the fMRI data from human brain, which is non-invasive. So no matter what kind of format neural data you get, these are just the current ways that we can collect the data from our brain. And then these neural data or neural activity may generate a certain kind of behavior. Right? So this is a very standard kind of diagram with the sensor stimuli, neural data, and behavior. And we put it onto two sides because we want to understand, for the neural coding problem, we usually want to understand how this neural data encode the input stimuli, which is observed, and this behavior data as well. Okay, so when we try to build a model to understand the functional relationship between this output variables with the input, with the internal neural data, is what we call encoding models. Okay, we want to understand what aspects of the neural activity really carry the information, how these neurons represent this external information. Okay, mathematically, we can express this way. We represent the external variable as x and the internal neural data as y. Okay, and building the encoding model means we want to infer the conditional distribution py given x. Here we say it's a distribution because we usually think the neural activity are not deterministic function of the external variable, right? There's a lot of stochasticity there. For example, if you present the same stimulus to the same group of neurons, you might collect very different, not very different, but some different neural activities due to the stochasticity or the noise, if people say. So we want to develop more flexible statistical models as this encoding model to understand the relationship from the x to y. Okay, so that's the kind of the motivation slide, what we are trying to do. We're gonna have this py given x show up a lot of times, so make sure this makes sense to you. Okay, so now the goal is to learn some function from x to y with some noise or stochasticity. Okay, so what we can do, the simplest thing we can do is build a linear model, right? Assume the external variable x is linearly mapped to the neural data y. Here, for example, x is the contrast of the images, okay? So that's a scalar. You can map the scalar to the neural response y using this linear model. Theta 1 is the linear weight and theta 0 is the intercept. Oh, question?
Yeah, can you give us a brief example of where the... Okay, thanks. Could you give like a brief example of where the stochasticity comes from? So why are the outputs of the neurons non-deterministic?
I think it's more due to the stochasticity of this biological property. Even you present the same stimuli to the same neuron, and there's some kind of neural circuitry level of activity happening, like the soma accumulate the input current, accumulate the signal spikes, and then depolarize and reaccumulate again. So these processes involve a lot of ion channel and open and close, and all these things are happening non-deterministically. So cumulatively, the whole, even like a single neuron level of activity of spiking event is not as deterministic as you expect. And then if you look over the whole population, the whole brain, nothing is really deterministically predictive. So, okay, so I hope that helped that question. And this is the single linear model. So far, we haven't introduced the stochasticity or the noise yet, but let's wait for a second. And a bit more complicated multiple linear model can be represented this way, right? Instead of just assuming the neural response is a function of the contrast only, it can be a function of multiple properties of the input stimulus, like the orientation, contrast, strength, a lot of properties, right? So, and we assume this response is a linear combination of all these stimulus features, and still keep the intercept constant there, because that tell you a kind of constant bias, right? So this is the linear regression model if you come from a machine learning world. And then you can rewrite this form into this vector form by stacking all the features x and the coefficient theta into two vectors. Okay, so y is theta transpose x. We know x, which is the input stimulus. We know y, which is the collected neural activity. I index for different neurons. I index for different stimulus, if you say. For the same neuron, it might respond differently to different input stimulus. So that's what this I index is for. And then you can further build it up to a matrix form by stacking all the stimulus together, right? So now you have a matrix X, which is the rows correspond to different stimulus, different stimuli, and the rows, the columns correspond to different features. Okay, so you want to infer the theta vector from the given observed x and y. So this is a pretty straightforward linear regression problem. And we haven't talked too much about noise. So here, if we assume the observation is from a Gaussian distribution, which means we want to add some Gaussian noise to this underlying linear model, then what you can do is for each single stimulus, you assume the observation, the neural response is generated from this linear mapping plus some Gaussian additive noise. Okay, that could be IID for different stimulus or different events, which means you can add a vector theta, which is the stacking of all this eta value. Each eta is an IID sample from the same Gaussian distribution. Okay, now you basically introduce this noise thing so that your observation, neural observation, is not always the same, is not always a deterministic function of the input stimuli multiply some linear way, but actually with some additive noise. Okay. Now, when you build this model, one thing I want to quickly mention is if you write down this py given x as we motivate at the beginning, this py given x would be a Gaussian distribution where the mean is at theta transpose x and the variance is the variance of the noise, which is sigma squared. Okay? Now the problem is we have a Gaussian likelihood, and then we want to infer the model parameter theta and probably the variance sigma as well for this Gaussian distribution. How are we going to do? The standard thing people do is MLE, which is maximum likelihood estimation. Right? You want to do MLE to maximize the log of the log likelihood of this Gaussian observation with respect to the model parameter theta. Okay, so nothing. So here we're showing at the top is basically if you have a Gaussian density, if you're familiar with that, and take a log, that's gonna give you that equation. Okay, where n index the number of 12s or data points or events, and d is the number of stimuli. Okay, and then very straightforwardly, you take a derivative of that log likelihood, set it to be 0. You will end up with a very nice closed form MLE solution, which is written in this way. Okay, x transpose x is all about the input stimuli, and x transpose y is kind of a reweighted sum of the stimuli by the neural events. We'll go through We'll go into more insights for these two parts later. Okay, so now we basically propose a linear model. And we solve the problem by fitting a Gaussian density function to the observation, to the neural data. And now we solve the problem. We know what the model parameter theta is. Okay, so you can say now you kind of already infer an encoding model py given x. This encoding model is a Gaussian distribution with the unknown parameter theta being calculated in this closed form way. Okay, problem solved, which means if you observe the neural activity from a single neuron, and you also know what's the input stimuli trigger this activity, you actually already built a model to understand the functional relationship between the input and output. Okay, any questions so far? If you understand this, and then all the things we do following just make this model better and better. Okay, because here we have a very naive assumption, which is the noise is Gaussian. Gaussian means the random variables should distribute from infinite, negative infinite to the positive infinite, right? There's no restriction over the value that these random variables can take. So also, the mapping function is linear, which is super naive. But actually, in practice, sometimes linear function really work well. So although it's naive, but it's really a practically effective method. Okay, so we have these simple assumptions, and all we need to do next is make these assumptions fitting the data, fitting the model, fitting to the real data better. Okay, so here let's show an example of using this linear regression model to the spectrum, right? Here the x is the stimulus, which are the stepping shape. and the y is the spectrum. And still, we want to fit the model. Y is theta transpose x. We know x and y. We don't know theta. And note that if this is all we get, right, the thing on the left-hand side is all we get, then we only have one data point, right? That seems to be not sufficient to fit a model. Then usually what people do is people actually truncate the whole sequence into multiple windows, okay? So what they do is they put a sliding window from the beginning, t indexes for the end of the time points at the end of the window, okay? And then you just generate some data points by sliding over the entire sequence. And the window size can be d, which is the number of features you include, right, as we show in the initial linear regression model. And the theta is still the linear filter or the linear weight. It has the length of d, and it will be applied to that specific window. And but you're going to have the same theta for all the window, but the xt will change as you slide it over the entire sequence. And let's denote this multiplication to be lambda t, which is a deterministic product of the two parts. Okay, we call that spike rate usually. Okay, you can actually rewrite this inner product as the summation over the scalar product. And note that here we actually flip this the order of theta with x. So x, if you go from the left to right, but theta actually go from the right to left. So, or what I'm saying is, when I is equal to 0, we're dealing with xt, and then theta 0 correspond to xt, and theta 1 correspond to xt minus 1, right? So you actually have a theta, you flip it, and multiply that with the x. People, if you're familiar with convolutional filter, this is very similar to just convolve a theta vector across the entire sequence. So, and the y value for each time point, which is like actually a binary event, right? Spike means whether the neuron bursts or not. You can think of it as a binary event. It's 0 and 1s. And it is the lambda t plus some Gaussian noise according to our previous assumption. So we assume the spike event, which is binary, from lambda t plus eta t, where eta t is still from a Gaussian noise. Okay, so we can actually walk through the data when increasing t and see how this sliding window happens, right? So this is the data, the first data point with the initial window and for that yt value. Then we slide to the next position, build the same linear regression model between x2 and y2, and then we do the same thing for all the t's. Okay, so that's basically the way to construct the data from this long sequence of stimulus and the long sequence of spike train. Similarly, we can reformulate everything into the matrix form, and we still have y is equal to x theta plus eta. Two differences from the previous simple example, where x are not just contrast orientation or this properties of stimulus, it's actually the sliding window over an entire sequence. So each row is a shifted version of the previous row, and y is a binary vector, 0 and 1s. Okay, so we can still fit this model to learn theta using the MLE with the closed form solution, which give us the same solution here, right? And x transpose x is what we call the stimulus covariance. Okay, that contain the second order statistics from the input stimulus. And the x transpose y is what people call spike triggered average. I think it's kind of intuitive because y is now 0 and 1s. You only average the rows if the corresponding spike is 1, right? So that's why it's kind of the spike triggered average. It's not just taking average, but reweighted by its spiking events. Okay, so this is a very straightforward and very simple question.
when you take the sliding window, I mean, you kind of lose the time dependency because whatever outside the time window, is that, does that matter for the prediction?
Yeah, so usually the reason we keep a sliding window is because we want to keep some temporal dependency. You can totally just grab the current xt without the history stimulus and use that to predict y. That would be the very extreme case where you lose all the temporal dependency. But you can change the sliding window. The longer, the more history information we'll get. But usually people assume like the spanking event encodes some history, but not that long. I don't think a single neuron can really encode say the history of stimuli for 3 hours ago. So it's actually reasonable to pick a window, not too long, but not too short. And this is like a hyperparameter I need to fix in the model fitting. But yeah, the reason we have the sliding window is because we want to include some temporal dependency, but not too much. Cool. Okay, so this is just applying the same question.
So this is related to the last question, but I was just wondering if the choice of D is usually just like whatever fits the data best, or if it reflects, prior assumptions about how much of the history is encoded.
So in practice, generally we do kind of cross-validation to see what's the best hyperparameter you can choose, because all these things are statistical models. They're all data-driven. We are not introducing any domain knowledge or any expert knowledge so far yet. So we definitely want to find a model that explains the data the best. But that not being said, you cannot introduce any knowledge you have. Actually, if you do know, how many times this window spans, and say like 10 milliseconds, and like these neurons don't really respond to anything before, you actually can introduce that knowledge. But actually, speaking of it, I think that could be an interesting thing to infer from the data that, actually this neuron respond to 10 millisecond window, the knowledge the best. If you go longer, it doesn't change the performance too much, or it might harm the model fitting. So that kind of gives you some idea about how long this specific neuron encode, how long the history the specific neuron might encode. So a lot of data fitting models will tell you you might have some hypothesis, you can try to test it out by trying to fit different kind of models to it to see what would be a more reasonable hypothesis. Yeah. So, but generally, when we fit the model, we kind of we've been working with simple neuron complex neuron in visual cortex, and we kind of have a rough idea how large this D can be. But yeah, that's more from the experience rather than some theory that we have. Okay, so this is really an application of the linear model to the specific stimulus-driven spiking events. Okay, and you can obviously see, assuming Y comes from a Gaussian model is not reasonable, right? Because Gaussian will assume real valued random variables, but here is literally just binary 0 and 1s. So the problem is the noise is not Gaussian. Okay, and we also see the neural response is non-negative. It doesn't take negative values. So we definitely need fancier models or more complex models in order to really capture the characteristic or the statistics of the observation and the neural observation. Okay, so let's summarize this model, linear model again using this diagram. You have input x pass through some linear function, theta x, add some Gaussian noise to it and generate the neural response, right? That's what we do. But generally, the noise usually people use more exponential family distribution rather than just Gaussian. So yeah, linear Gaussian model is one example of this. But sometimes people want to have more generalized linear model, which is literally the generalized linear model name. And the way to generalize that is after the linear mapping, we will have a nonlinear function, right? To allow it, to maybe more flexible and fit more nonlinear encoding models. And also the noise can be more flexible among the exponential family rather than just a single Gaussian distribution. A straightforward extension from the linear Gaussian model would be this nonlinear Gaussian model, where you put a nonlinearity f outside of this theta x product. People call this f to be in the nonlinear function or the inverse link function. I'm not sure why, sorry, the f inverse is what people call link function in computational neuroscience. That's just probably some convention from the early literatures. Okay. Another variant, which would be more practical, is instead of assuming the observation y's from a Gaussian distribution, you can assume y's from a Poisson distribution, where the input to the Poisson is the f of theta x. Okay, so that's now we generalize the simple linear Gaussian model to the nonlinear Poisson model by introducing the Poisson distribution as well as a nonlinear mapping function. Okay, and we actually can show in practice that this is a more general and more feasible encoding model for spectrings rather than the linear Gaussian model. Okay, any questions so far? Question?
Thank you. Yeah. So this F, is it something that is fixed beforehand, or is it something that needs to be learned?
So usually we fix it for simple models. I will show later what the F we're going to use, but you certainly can make it more complex, which I will also show in the future slides, like what a complex F you can do, you can assume by learning, which requires a model fitting or parameter estimation. So. Yeah, so usually when you collect this neural data, like the spectrings, people treat it like so that that will relate to the how the single neuron behave. Single neuron take the signals or current from the upstream neurons, and the current is like the voltage, and they will be accumulate. As soon as it passes a certain threshold, it will burst, it will generate kind of burst event. After it bursts, it releases the signal or release the energy, it will kind of drop down to a very low level membrane potentials, that's what people call. And then the neuron becomes silent. And then actually there is a refractory period where it's just silent, you cannot retrigger that again. And then when it bounces back to the normal membrane potential level, it will keep accumulating the signals, input signals, and burst again. So that's how a single neuron performs. So if you collect the single neuron activity, you will likely see the burst events like bzz, bzz, bzz. So that's what people call spikes. And that's usually like the binary. So actually when you see the raw data collected from the electrophysiology experiment, there are actually time steps. So like 3.2 milliseconds, there's an event. In 10 milliseconds, there's an event. So that's what you see from the raw data, and you convert that into a binary event. Say at that specific timestamp, there's a 1, otherwise it's just all 0. And people actually, I will also show later, is it's actually more challenging to deal with these continuous timestamp because people need to assume that's come from a point process, which is harder to fit because of the likelihood in the point process. So they have to convert that into some simple distribution like Bernoulli distribution or Poisson distribution. Poisson is actually, you bin that, right? For a specific time point is a 1 and 0. But let's say you use a bit larger time bin to binarize your whole sequence, you might get multiple counts in a bin. But this multiple counts is still an integer, so people use Poisson to, yeah, that's why it's Poisson. So that's actually come from the raw data, which is continuous point process, to the binary events to the Poisson distribution. Does it make sense? So for the so actually people, if you consider that's binary events, people use Bernoulli and logistic regression. So yeah, I mean, I think logistic regression is actually Bernoulli distribution over the posterior of the Y. So essentially, I think you're right. Like people, when you say logistic regression, you're essentially using the Bernoulli distribution to model your neural response. So, but actually, nobody really used logistic regression because they don't treat them as a classification problem. Yeah. But essentially, they're the same, I would say. Yeah. Questions on this side?
Quick question for the Poisson model. So in the Poisson model, what's the form of the eta moister? Do we consider as approximating the firing rate and keep the mean and the maximum the same, or does the eta doesn't depend on later?
So the eta and f has no relationship. You can have modular. It's actually, I would say this form of GLM is modular, right? You can swap in different parts for different stuff. So, and interesting from Poisson model, there's no way to write out eta explicitly. You just, because for the regression Gaussian model, you can just also say y is from a Gaussian with the mean as the f and with a noise variance. So this way of writing out the eta is actually more specific to Gaussian. But if you have more general distribution like Poisson, like Bernoulli, you don't have to explicitly write out eta. You just use f theta x as the model parameter of the density function of that specific exponential family distribution. And here, that actually, I showed that early. So f theta x is what people denote as lambda, which is the spike rate. I will show in the next slide. And lambda for Poisson distribution and variance, the mean are the same. So the mean and variance of Poisson are both lambda. And that's actually, people also have later expansion. What if they have larger variance and smaller variance, which correspond to the over-dispersed and under-dispersed Poisson model to model the neural data? But this is a very straightforward, a more straightforward model. Question, yes.
So here, y instead of an array of binary values, it is actually the count of spikes within some time window. Is that
Yeah,
right?
exactly.
Oh, okay.
Yeah. Okay. So when your time bin is super small, like it's smaller than the smallest interval between two events, then you will get Bernoulli or like binary. But as long as you increase the bin size, you might easily collect two events in one bin, then you will have Poisson. So that's exactly the number of counts within the bin. That's correct. Question?
So is that like saying that we're usually more interested in the firing rate as opposed to the like specific sequence of zeros and ones? We're more interested in like how often the neuron is firing in a particular unit of time?
Right.
Okay.
So, but the thing is, we only have y, we don't have lambda. So what people usually do is they want to infer lambda from the y, and lambda might tell you more continuous property of the neuron, which is, I wouldn't say not more interested, but like easy to be modeled, right? It's easier to work with continuous rather than discrete values.
So is there a model where the noise is within the link function and not outside? Like, you know, in
I
signal
think you
processing,
can. The model I'm introducing is very basic. It's like the intro level class. But I think for people definitely put a lot of effort research in making this model as sophisticated, as stochastic as possible. Actually, I think I'm going to talk about something like before you pass through the X itself. Well, that would be related to the latent part. But yeah, whatever inside of F can be also stochastic. The model can be as complex as you can imagine. But this is a very, actually, let me just jump to this slide. I mentioned it as a basic Poisson generalized linear model because it's literally just the basic model. And so it's actually the same as the previous equation, just like visualized with the diagram. You have a stimulus xt multiplied with a filter or with a linear weight xk. So sorry, I changed the notation here. K is theta in this previous slide. F is here. An example is like exponential nonlinearity to answer the question that F would be exponential in this case, but you can certainly use a sigmoid, ReLU, soft-plus, tangent. So similar to a neural network, right? Build neural network, what kind of nonlinearity you can use. But if you use this kind of simple nonlinearity, you are not assuming any parameters. They're very just straightforward nonlinear functions. And then output is lambda, which is spike rate. It has to be non-negative before put into the Poisson model. Right, so that's why we put an exponential. And all the functions I mentioned, except for the tangent, all the others give you positive or non-negative values. So you want to, you want to map the linear product into some non-negative space using this nonlinear function, and that is input to the Poisson spiking model to generate the Poisson events. Here, actually, because it's not really precise because I'm showing y as the binary events, it should be using Bernoulli if you want to be more precise about this procedure. Okay, and a more explicit way of writing out is the spike rate lambda is f of kx, and the spike count y is Poisson of lambda. People also call this as a cascade model or like a LNP model because you have a cascade of linear, nonlinear function. And when you say cascade, you can easily think like, what if we have linear, nonlinear, linear, nonlinear, right? Like LLN, right? That's why people call it cascade. We're actually going to show an example of LN model later. Okay. So yeah, this is a very basic GLM with the Poisson distribution and with exponential nonlinearity. And you can easily fit this model and the linear Gaussian model we showed at the beginning to the same spike train data, and you will see this will explain the data better. And this will learn better stimulus filter than the linear Gaussian model. K is the only unknown parameter in this mapping, in this encoding model that you need to infer, and you infer this using maximum likelihood. Question? No? You're trying to raise your hand. Okay, so yeah, so that's another simple basic model. So what we can do beyond that. One thing we know that neurons not only encode the history stimulus, but also some of its own history events, right? If you already spike how many times, and the neuron know that, and we'll take that information for the future events as well. So one straightforward extension is to incorporate the spiking events or spiking history. So people introduce this spike history dependence Poisson GLM by putting the output spikes back to the input of the exponential function, and the math is written out as here. You can think of it consists of the two parts. The first part is the stimulus. The second part is the spike history. Okay. The only thing is right now this output is not just a Poisson process. What do we mean by Poisson process? Poisson process means the arrival of the events is independent of the history events, right? Clearly, we introduced the history events as a part of the model generation, so this part is broken, right? So it's not really a Poisson process. People actually, so I think this is more like a hockey process, if you know more about point process model. So like hockey process is a kind of self-exciting process. You use your own history spike events to excite yourself. Okay, so that's what actually people call hockey process. And interestingly, this kind of model is very similar to a theoretical neural model called integrate and fair model from very theoretical neuroscience. If you come from theoretical neuroscience, you probably know this integrate FAIR model, which describe how a single neuron integrate from the history, current, or event and FAIR. So GLM, though, is a pure statistical model, but by constructing the model this way, it can mimic some behavior from some theoretical neuroscience models. So here I want to show a bunch of examples. By designing this H, which is people call a post-spike filter, you can achieve very different spiking pattern. The stimulus filter is the same, which is that like Gaussian bump with a longer tail. That's always the same. Which is the K, right? You pass K through the sequence of the stimulus, and then you will generate the filter output, which is K multiplied X, right? That part is unchanged. Now the spike rate lambda is a summation of that part plus the post filter multiplied the spiking events. Here the post filter is 0, so this is just the regular, the Poisson GLM without any a history dependence. And you can see you just get random Poisson events or spiking events, right? But as I mentioned, there's actually a lot of interesting dynamic patterns or dynamic behavior of a single neuron. For example, some neuron might just spike very regularly when triggered by a certain stimulus, right? Like regular means you have pretty equal distance between two events. And you can actually design the post-spike filter like that way to achieve such a regular pattern. The intuition is like what I say. I mean, a neuron, after it bursts, draw back to a very low membrane potential, and it will stay there for a while, which is the refractory period. During this period, if you give it the neuron very strong signal, it won't spike, right? Just stay there. After a while, it will, recover back to the normal membrane potential and allow for more spiking. And that post-spike filter actually kind of mimic that mechanism, right? Right after the spiking event, it will have a very deep inhibitory kind of signal to say you should not spike after you already spiked once. And then that inhibition will kind of decrease and recover to the normal level. Now you can go back to the spiking events again. So this will give you a very regular, equal space spiking events. And then you can also design the post-spike filter that way so that you can achieve something called adaptation. So you will have a very strong inhibit signal right after the spiking event, but then bounce back to a very normal level of membrane potential and slowly adapt to the normal, the true normal level of membrane potential, which give you a lot of bursts, very dense bursts at the beginning and gradually become a normal Poisson process. Also, this is another kind of interesting events that you burst equally, no bursts, burst equally, no bursts. Okay, so some neurons exhibit this kind of dynamic properties or behavior as well. Weber and Pillow paper 2017 actually show you can design GLM a lot of ways by having different stimulus filter and post-spike filter. Then you can achieve different dynamic behaviors that are very similar to this, this theoretical neuron model. The way to do that is actually very simple. You have the theoretical model generate these spiking events, fit the GLM by learning the H and K, and see what is the best K and H. And the red is the H and the K is the blue is the K. So that's what you learn. They can generate these kind of spiking pattern from the input stimulus. Okay, this basically show a very flexible, the very strong flexibility of this pure statistical data-driven GLM model to generate the different patterns that is very similar to the the theoretical models, and can capture different patterns from the real neuron activity. Okay, so this is just to show like GLM is kind of powerful in this sense. Any questions so far? Okay. Next, we are also interested in further extending the GLM into a multi-neuron GLM, because one thing we know that neurons talk to each other, right? They send signals to each other. They're not just independent identity. So a straightforward thing you can do is you just input your spiking event to other neurons, right? So now you have this coupling filters. You don't just take the history of yourself, but also the history of other neurons. So that's a very straightforward multi-neuron extension. If we write this with the standard GLM diagram, you can see for neuron 1, right, Y1, that yellow highlighted spike is actually generated from the stimulus, its own history, and the Y2 history. Okay, writing out the math, it would be Kx, which is the stimulus, and the sum over all the J. J indicates the neighbor neurons, but J can be itself, can be I again. So this part is tell you how different neurons interact and communicate with each other. So this is actually a very flexible, more general type of multi-neuron history-dependent GLM people usually use to fit today's question.
Thanks. Just out of curiosity, but what ballpark magnitude is J? Like how many neighbors does a neuron have? Is it like 10 or like a billion?
How many neurons you're interested? It depends on your data sets. If you have 100 neurons collected, then 100 neurons. So because we might assume a fully connected graph, I'm not sure that's a too buzzword, but if you think like all the neurons are fully connected to each other, but then you fit the H, right? So H is learnable. It's possible that you learn H, which is just a zero vector. So that means this neuron is not talking to that neuron. But if you learn some sensible H, that means they do have a talk, and in what pattern one neuron sends signals to the other neuron. So usually it basically depends on how many neurons you have. People fit from 10, 7 to 100 different scales. Yeah.
Okay, thanks.
So this accounts for like, you know, two-way interactions where one neuron affects another and another one affects the same neuron, but does it account for like 3-4-way interactions, or is that a different, more complex model?
So basically, this is a pretty complex one. You can think of one neuron gets signals from all the other neurons. And this is true for all the neurons. So if you have 10 neurons, so I will show actually in the next slide an application to a retinal ganglion cell population. We build, for each cell, you build this model, but you also build this model for all the cells. So this is, you can think of all by all connection, all by all talk to like communication between neurons from the entire population. Does it help answer your question?
Yeah.
Okay, great. Cool, that's it. That's all the models I want to introduce so far. Like maybe a lot, like we talk about the simple Poisson GLM, we talk about the spike dependence, and also the multi-neuron one. So here is one example. The stimulus, the binary fully rate, just 0 ones. Or if you shift it, it's from -0.4 to +0.4, okay? And then you can collect the spike train. Actually, here is the spiking time, which is the continuous time step. And then we binarize that into the spike counts by fitting, I'm not sure what's the size of beam, but rather it's between 2 millisecond to 20 millisecond-ish. And then now you have a spike counts, which you can use Poisson to model that. Okay, so go back to the early GLM we introduced. You first formulate the design matrix, which is X. And if you see, it's actually literally the shift version of the window, right? You slide the window across the whole sequence, and then you will have this. This is actually a topless matrix. And then Y is the spike train, or is the spike count event sequence. And you want to learn the encoding model Py given X, right? And using the Poisson GLM we introduced. And then when you have the spike history from its own events, then you just attach a spike history portion that is formulated from the history of the spike response as an extended version of the matrix, okay? But the model fitting is actually the same. Although we introduced different math, right, to fit the model, but essentially, when you formulate everything into the matrix version, it's actually the same, which means you just change different acts, but the encoding model is still just the Poisson distribution. And then you can also have the different neuron spike history formulated as the extended matrix to attach to the original design matrix. Okay. So this is just a slide about fitting the model. You have the data X, which is a tensor, if you have image rather than a scalar. And then you have Y1 to Yn, which are the spike count, the spike response for all the neurons. You want to infer K, which is a tensor, has the same dimensionality as the X, and all the history spike features from all the neurons. Okay, and then write down the log likelihood of Poisson distribution, maximize that with respect to all the K and H. The good thing is this likelihood is concave, so you always converge to the global optimum. That's why people actually like this kind of model, because it really has this nice optimization properties. Here is one example applying the multineural response to this RGC, the retinal ganglion cells. Each circle or each ellipsoid is one cell in the population. And here we show 11 cells. You can see the dashed line is the real neural correlation that you calculate from their real response. And then you can fit a GLM model with the multi-neuron correlation, or just a plain GLM without multi-neuron correlation. You can see the red curve fits the dash curve better. It means if you assume neurons talking to each other and infer this spike history age from different neurons, you actually explain the data better. Okay, so this basically show that there exists strong correlations and communications among these neurons in this RGC population. Okay, so far everything makes sense. Cool. Actually, I have one last model I want to introduce on the GLM, then we can move on. So, one more GLM model I want to introduce, which is the more complex version, is called the subunit model. It's actually a LN model, right? You have two layers of LN. The first layer, different part from the previous, is the filter doesn't just multiply the X, it's actually convolve over the X. So this part is very similar to CNN, right? You have a convolve filter over the entire image rather than just have an MLP where you multiply the whole vector of weight to your image. So this part is a convolutional filter, subunit filter K. But when each time you multiply with a certain portion of the stimulus, you generate a scalar. That scalar will pass through a subunit nonlinearity F and generate a negative scalar, and then multiply, and now you have a vector of scalars, right, from each position of this convolutional operation. And then you linearly combine them together with a weight, linear pooling weight, W, to generate one final scalar pass through the G, the output nonlinearity, to generate a spike count. So this structure, the mathematical formulation is on the right here. But if you, as I go through this, it might give you some familiarity if you're familiar with convolutional neural network. You first have a small patch convolve over the entire image, generate scalar for each position, do a max pooling or average pooling, then finally you get a smaller tensor, right? And then pass through the next layer. So here the idea is similar, right? You convolve first and you pool. But here we assume a pretty strong form of structure, and the output of the model is a scalar rather than some smaller size of the tensor. Okay, this is actually inspired from this retinal subunit model, from this rod and rod of bipolar neurons in visual cortex. So this rod of bipolar neurons, which is actually after the sum, take the information from multiple rods and pass through some nonlinearity and take a summation. So this is from some kind of very neural physiology kind of experiments, and summarize this might be the computation model under these rod neurons. So inspired from this kind of operation, people come up with this subunit model. Different from the traditional GLM, it's more biologically plausible because of the similarity to the real neural structure, and it's more parsimonious. Parsimonious means instead of having the whole vector for the entire image, you only use a small filter and convolve, right? So that's require a lot less parameters to fit. But it's actually very computationally powerful. It can fit very flexible models and learn very good, can explain the data very well. The main issue is we cannot just fit a maximum likelihood to the Poisson distribution and get a really nice concave or convex objective landscape. So it's not easy to fit to the data. And this is from my work that we actually we propose a new fast-fitting method using moments method. And we show some theoretical connection to the spec-triggered average and the covariance analysis, which I mentioned very early, right, with the closed form of the linear regression model. We show the proof of model identifiability, which means the parameters we learn is identifiable up to some constant shift. So I just want to quickly mention that the thing we do is actually straightforward in the sense that we first have this general spike rate formulation for subunit model, and then we have two strong assumptions. Well, not that strong in the sense that we will show is actually okay if the model has if the true data has some mismatching properties. But here we assume G, which is the output nonlinearity, to be exponential, and F, which is the subunit nonlinearity, to be quadratic. Okay, if you assume these two nonlinearity, then rewrite the subunit model, you will get something called generalized quadratic model. So GQM is actually some early extension of GLM by extending the linearity into a quadratic nonlinearity. And we actually make a connection between the subunit model and GQM by assuming these two nonlinear functions. And the lambda is the CX plus BX plus A, that's the general form of the GQM. And we show the C matrix in GQM actually has the form of K diagonal WK, where K is the topless matrix of the subunit filter, and W is just the linear pooling weights. And B also have a connection to WK. And because of the special structure of these two C matrix and the B vector, we actually do all the theory and proof thing in the Fourier domain because of the convolution, because of the topless matrix structure. I'll skip that. But the cool thing I want to mention is, if you write down the log likelihood of the subunit model with this quadratic assumption, you will end up with the form like on the third row, right? So you have two yellow blocks and one green block. And the one green block is actually very time consuming because it needs to be calculated over all the data points, right? If you have super long recording, then this I, sum over I, is a huge consumption of the computational power. So one thing we introduced that we can replace that giant term, green term, with something called expected log likelihood. So we basically approximate that summation using an expectation operation. And by doing this, you can actually get a closed form second order statistics, also the first order statistics from the model. So now we basically have a very easy, fast calculated term instead of taking a very cumbersome summation over all the data points. So that's kind of the main trick we introduced to speed up the inference procedure. Anyway, here is one simple simulated example to show if we know the true K and know the true W, because we simulate the data and we use different models to fit to the data. And our model, which is the so yeah, our model can basically get pretty nice estimation of K of W, but compare with MLE, which is calculating this cumbersome, like very heavy block, grain block term, it's super slow and increase exponentially with the data size. But our model keeps constant because we don't require to calculate that term. We applied our model to the real data sets from the V1 complex cell. And we know its input stimuli are this 2D spatial temporal white noise. And we get the neural response from complex cells. And then we will fit different subunit models using our method. And we show that our model still stay constant in terms of the running time, but the model fitting performance is very high, close to this very accurate MLE estimation. Okay, and finally, we show that this two subunit model means we actually fit two subunit model to a complex neuron. The top row is the true statistic of the real neurons, and the bottom row is the statistic of the predicted neural activity from the subunit model that we fit. We show like just by fitting two parameters, K and W, we can already recapitulate the statistic of the real data. So that shows how parsimonious the parameter space is, but it can capture the full statistics of the real neural response. So it's a really computational powerful model. Okay, so quick summary is we start from a very simple linear model, extended with some nonlinearity and a fancier noise model. We introduce the spike history and show how that can capture different dynamical properties. And then we introduce the multi-neuron correlations. And finally, talk about the subunit model as this cascade of LN models. Okay, I'll just pause here for the GLM, but also happy to answer questions if anyone have.
I have a very basic question. So the final aim here is to come up with a model which is more biologically plausible, or any model that would explain the data.
I think it's more biological plausible, because the more biological plausible, usually the better you fit the data. For example, if you have a Gaussian noise and Poisson noise, likely Poisson fits better because we know the spike events are more kind of discrete rather than continuous. Maybe that's not biological plausible enough, but it's kind of a one step towards the true statistics or characteristic of the neural response. So people also fit a very, like, throw a neural network there, get pretty nice results. That's also fine, but generally I think computational neural people or neuroscientists don't like that because they're less interpretable. Actually, there's a lot of interpretation going on among this model. Like, people can look at this filter, like stimulus filter, use that to explain how each single neuron responds to the external stimuli. You can see like Gabor filter, right? Like, you can also see some neurons has more respond to more one type of color or like one type of orientation of moving stuff. So you're easily able to interpret this kind of thing from the simple model. But if you just fit a neural network, get some reasonable prediction for the data, but how are you going to do that beyond that? more not for really scientific discovery or scientific interpretation.
One more question. So how do you recall? How do you do the inference of this theta? That so how do you recover the model? Is that coming into like how what algorithm? Like in some of the cases, these are convex programs, but as you have more non-linearity, I'm pretty sure soon it is going to be non-convex.
Yeah.
So
Basically, for this type of model, people use maximum likelihood estimation because all you want to do is py given x.
but the likelihood may not be a convex function, right?
Yeah.
So
then you need to pick good optimization tool, which is outside of the scope of our research. We hope people can develop better optimization tool that we can grab and use and to solve the non-convex problem. Yeah.
Thanks.
I think this is supposed to be a break session. So if anyone want to use the restroom or anything, want to take a break, it's okay. Please go, yeah.
Yeah, could you go back a few slides to before the subunit to where there was all the correlations in the matrix?
This one?
Yeah, there. Or,
yeah, maybe stop here.
Yeah, right there. So what I was getting at earlier, I guess, is that this model, fits to the data of like these and choose two correlations between any two neurons. But would you also ever try to fit it so that it accounts for like the n choose 3 correlations between like sets of 3 neurons or sets of 4 neurons?
I'm not fully understanding your question. What do you mean by choose correlations?
Oh, I just mean like, there's correlations between each of these two cells in the firing, but you could also account for correlations between sets of like 3 neurons.
Okay. Yeah. Group of correlate. That's a good question. For this kind of model, people treat each single neuron as a node in a graph, you would say. So people try to understand single neuron property. But you can, I don't know, like if you focus on the group of neurons, that's more, it's also hard to say in a sense that, actually I don't. So these days, people start to work with multi-region neuroactivity, where they will have neurons in different regions, right? And then they will build this GLM type of thing within the region so that each neuron will be one node. Now they will zoom out a little bit. They will make connections between the group of neurons or one region of neuron to each other. But I think the study in that kind of direction is still pretty preliminary so far. And one thing people do is they assume one connection from one group to another group. And then really not too much insight or too much very insightful ways of building connections between group of neurons. I'm not sure that helps to understand that question.
So can we go to the summary slides again?
Okay.
So here, so for the first slide, are we saying that we need to apply some dimensional reduction methods to our X first, and then fit the linear model, and then wrap that around the nonlinear model? Is that the case, or do we just?
Oh no, I think so far people don't do. I think when I say dim reduction, I actually adapted slides from Jonathan Pillow's tutorial early. The point is more about, instead of now, like for the whole X, I map the whole X to a scalar by mapping a K with it. So that's what the dimension reduction here.
I see.
Actually, in the next slide, I will talk about what is the real dimensionality reduction we're gonna do.
Yeah, thank you.
Yeah.
So once you have a model and you want to evaluate it, is the basic way the likelihood? Is that's going to be the quantitative
you split
way?
the data into training test and fit the test likelihood.
Okay, thank you.
Yeah.
Thank you. Thank you for the talk. And I have one question. That is, when the two neurons have some like high covariance in their activity. That might because they have a connection, and that might also because they have some shared inputs. And I'm wondering if there's any efforts in like explain away the shared inputs, but only predict the true connection.
That's a very good question, and that's unsolved so far in the field even. People try to connect anatomical structure with the functional connectivity. I think so far what we can do the best is try to understand the functional connectivity, or people call effective connectivity, really how neurons functionally affecting the others. But how that information relates to the anatomical structure is different in different cases. But we do have a common sense that the anatomical connectome might regulate your functional connectome somehow. So we actually just recently work on something to use that information to regularize the dynamically changing functional connectivity among neurons. So that is, that hopefully that kind of answers some part of your question regarding to the anatomical part, or how they really connect to each other. And another part of your question is like two neurons actually relates to each other might through another neuron, right? That's actually a very hard causal problem. I don't think anyone solve it in neural population or this kind of understanding. But one thing people also try to understand is, or try to build a model is what people call partially observed point process, which means we certainly don't assume we collect all the neurons. We only collect partial neurons. Then what if there are some neurons that don't collect, but they actually affect a lot of observed neurons as maybe the common like confounder or something. And then we do develop like this partially observed hockey process and stuff, try to get a sense like how we can really understand some hidden unobserved neurons, or maybe group of neurons, and how those affect the observed neurons using this point process or this Poisson process models. So that's a very, not very, but some more sophisticated topics people study as well. Yeah, but that's really good questions.
So we have a couple questions online.
Okay.
Do features X need to be linearly independent in most cases to interpret the learned weight matrix properly? For the cross-neuron model, the firing rate between the neurons can be correlated with each other. How to interpret the weight of the model then?
For the first one, it doesn't have to, but like if you fit a linear regression model yourself, you definitely want to have some more independent handcraft features, right? If your features are very correlated, it might be less interpretable in a sense. But that's something, yeah, we don't know much, but we can try the best to learn some, to handcraft some features so that again, as independent as possible, and then so that we can know, because if two features are really correlated, then likely you fit one, you change one, the other one is going to change. So you might see some interesting correlation pattern as well. So that might help you to further reduce the correlation and understand the how each feature affect the neural response. So that answer. What's the second question here?
The second question is, what can we do using the model after we achieve good fit?
Okay, yeah, that's what I was saying about interpretation. I think people want to interpret. The goal is try to understand, as I mentioned at the motivation slide, right? How each neuron represent the external stimuli, right? That's actually one term called tuning curve, which is a kind of function for each single neuron. It should have a tuning curve. Tuning curve characterize the relationship between the input stimulus and the output neural response. So I'm not showing here. For example, you have the input as the orientation from 0 to 180, and then you should have like some curve showing like at which orientation this neuron fares a lot. And this kind of information should be readable from the inferred future of the GLM model. So the goal is to understand how individual neurons encode the external information. And using this kind of linear, but not that linear because it's generalized linear model, you can pretty well interpret them compared with a very sophisticated model.
Great, thank you. Okay, we will go for a quick 5-minute break and
Okay.
then we'll be right back.
Okay, cool.
All right, we're about to get started online.
Okay. Shall we start? People online, okay, can hear? Okay. Cool, cool. Yeah, I probably a bit slow in terms of the progress, but I do get a lot of good questions actually from the first part. And as I mentioned, the first part is actually, you can think of the basic thing, and all we're trying to do next is just to make it fancier and more flexible and stuff. So I mainly talk about the GLM, which is generalized linear model. It is used to understand the encoding from X to Y, right? Py given X. We assume we know X, we know Y, but we don't know the mapping function. But we also assume the mapping function is stochastic. That's why we use Py given X. There's another set of questions where we go deeper or more sophisticated is, what if we don't know X, right? We can collect a bunch of neurons. We don't know how these neurons are generated, like what really triggered these neurons, right? Because the GLM are very generally applied to visual cortex, where people know there's an input stimulus, and then we collect some V1 or V2 neurons, right? We know there's should be a mapping function. But if you collect neurons from, say, hippocampus, from prefrontal cortex, from motor cortex, right? A lot of, not just in the input part, but a lot of higher order processing cortical areas. We can't just simply build a GLM. And also, what if you want to understand not just a single neuron's behavior, but a population of neurons' behavior? So it's a very complicated encoding model to build. And in that case, people will extend GLM type of model into a latent variable model. Basically, you assume X is unobserved, right? It's a latent variable model. There exists some X that generate or trigger the neural response, but I don't know what these X is. So that kind of come to the second part, which is the latent encoding model. We still have Y, but we don't have the X. We don't have the observed covariance. And we still want to understand Py given X, right? But there's one more thing we want to understand is, what is this X, right? This X becomes the latent variable rather than the observed. And because the whole thing is based in statistical, and you usually put a prior over X, right? The simple thing is X is from some Gaussian standard normal distribution, but you want to kind of regularize the parameter space of X, which serve as this Bayesian prior. Okay, so now we want to build this latent model, which is the Py given X. People also call that data likelihood, because that's the observed data Y given some unobserved latent X, and multiply the prior. So we want to know the encoding model as well as the latent X, which using Bayesian language would be the Px given Y, right? The posterior of X given the observed Y. Using the Bayes theorem, you can tell Px given Y is this joint distribution divided by Py, right? Py is the marginal of the data. So essentially, in many latent variable model of this neural data, the goal people want to do is do posterior inference. People want to learn what's the best posterior distribution of the latent given the observed Y. But meanwhile, you also don't know the encoding model, which is the data likelihood. So you also want to do data fitting by learning the best model parameter that explain the data generation from X to Y. Okay, so we want to capture this hidden structure, and there are mainly two types of hidden structure. One is some low dimensional continuous trajectory, which I'll mention more in the next slide. When I say low dimensional, is because the neural data you can think of as high dimensional. If you have 1,000 neurons or 500 neurons, that's a super high dimensional neural space. And you want to map them to some low dimensional space so that it's more interpretable and more condensed. And there's another type of latent, which is called discrete state. Discrete state can be pretty low. It's kind of just a scalar. We will also mention this later, but generally, I will motivate the whole latent encoding model from the low dimensional continuous trajectory perspective. Okay? So here's the problem of formulation. We have the high dimensional spike trains. It doesn't have to be spectral. It can be arbitrary neural data, but here's spike train as an example. The y-axis corresponds to different neurons, certainly high dimensional. The x-axis corresponds to the time steps or the time bins. And we look at this and ask what these neurons are doing. No idea, because so high dimensional, there's no clear statistical patterns. So people usually want to find a low dimensional manifold or structure or trajectory that can explain these high dimensional spike trains. This exists. Actually, this has been, so latent here means hidden or unobserved. Actually, let me, so, okay, the question is whether this exists, right? Whether we can find some low dimensional structure. These high dimensional data does exist or lie on some low dimensional manifold. The answer is yes. It's very intuitive because neurons, you have 100 neurons, they're not doing 100 different things. They're actually doing some less than 100 number of things, right? So that means correlation. Neurons have strong correlation, and correlation means there exists a low dimensional subspace that you can, for each dimension, it might capture correspond to one correlated neurons, right? So one group of correlated neurons. So that kind of similar idea of why people do PCA or the other dimensionality reduction. So as long as you know neurons have correlations, then there should be some low dimensional structure underlying that. Actually, there are some existing research across different cortical areas have shown that they can find some really interesting interpretable low dimensional trajectories or manifolds underlying the high dimensional neural populations. Okay, so things definitely exist, and this is actually a very prosperously developing field in recent years. GLM is more like older, right, fashion, and in the recent 10 years, the LVM is more like prospering, developing. Okay, so back to the Bayesian language, to the statistical models, we have some latent X. We want to put some prior PX over the latent X. We also want to learn the probabilistic mapping or the encoding model PY given X. We don't know the X, we don't know the probabilistic mapping. Okay, if we can know these two, now we can say sample a X from the prior, map to the neural data using the probabilistic mapping, so that we can get this vector of Poisson for, say, a certain time point, right? So that will be the generative model that we can build based on these two probabilistic components. And then finally, you can write down the marginal of the data by doing the integral over the joint, over the latent, right? So we can marginalize over X to get the marginal distribution for the data. And if you're familiar with MLE, right? So MLE means you need to, we still want to do similar thing as GLM. We want to maximize the likelihood. But in the GLM, P given X is a full likelihood. You know X, so you don't need to marginalize over X. Here, we don't know X. We need to marginalize that and to get the final marginal PY. And this integral is usually, we want to maximize this PY to do MLE, right? So that's the standard thing. But generally, as you can probably easily tell, this integral is non-trivial. You're not likely to get a closed form integral solution. So most cases, people develop some approximate inference approach to do the, to estimate the model parameter, but essentially they're trying to, the ultimate goal is trying to maximize the marginal probability. Okay. Because I keep saying there are two different components, the prior part and the mapping part. And as I mentioned, people are really investigating this type of problem a lot. That triggers a lot of models, right, to solve this kind of problem. We can also think this as modular, right? The prior part can be one thing. You can impose different assumption over the latent, and the probabilistic mapping can also be various, right? You can assume different encoding models. So here I'm list a bunch. I'm gonna basically go into two different directions given all these modular components. You can put the px as rows and py given x as columns, and that will give you a bunch of literatures using different components to build different models. In the blocks highlighted in gray color is something I'm going to talk about first. And the common sense of these models is they all use some kind of dynamic assumptions. They assume the latent x evolve through the time following a certain dynamical system. So these are kind of the commonality of all these green boxes. And I'm also going to talk about the others, which is actually along the bottom part, which is based on the GP Gaussian process. Okay, so let's start from the top. You can see like the rows correspond to the green boxes are like dynamic system, recurrent neural network, and with some neural network or linear, nonlinear Poisson as the encoding model. Okay, so the simplest one is this linear dynamical system. A linear dynamic system assumes that the prior px is actually follows a linear dynamic plus some Gaussian additive noise. So for the first time point x1, it comes from some Gaussian distribution. And then for any following time points xt, it's only dependent on xt minus 1. But following this axt minus 1 plus b. So that's the linear dynamic part. Okay, so a neural population start from time 1 and then evolve across the time, right? So that's what we observe as the neuron by time matrix. But we assume there's an underlying dynamic generating these neural response across t time steps. And then if you treat each time point x1, x2, x3 as the latent as the xt, x1, x2, x3, then they assume the latent at each time point underlying the neural population evolves with axt minus 1 plus b. Here, because we keep saying we want noise, so we put a Gaussian noise, which is eta, added to the equation, which give us this the second Gaussian distribution. Okay, but the first one is called initial distribution. The second one is the transition probability. Okay, if you're familiar with Kalman filter, this is literally just Kalman filter. And then the observation y is another layer of noise, right? Because now if we say we already know x, right? X is generated from some Gaussian prior following the linear dynamic system. Now the y given x is just a linear Gaussian encoding model, right? As we introduced at the beginning. So py given x is simple, and px is just a linear LDS. I'll use the acronym LDS plus the Gaussian noise. Okay, and you can see there are some model parameters c and d are not, or c and d correspond to theta in the first slide of linear Gaussian, and a and b are the dynamic parameter of in the prior, and r is the noise for the Gaussian observation as well. So these are the set of parameters we want to infer by fitting the model. We can write down the joint probability px1, right, multiply the transition over all the time points and the emission, actually, or the encoding model for each time point. So if you're familiar with hidden Markov model, this is exactly the same as hidden Markov model. You have emission probability, transition probability, and the emission probability. Okay, so again, the linear dynamic system is actually the Kalman filter, which is the continuous version of hidden Markov model. People also call this Gaussian LDS, but by general, if people say LDS, then we assume the observation noise, py given x, is a Gaussian distribution. Okay, and then in order to fit to the model, all you do is get this joint distribution, marginalize over x so that you can get py, and maximize py with respect to the set of parameter theta, and that will give you the optimal parameters that fit to the data the most. If you're lucky, you will end up with a closed form PY. If you're unlucky, you don't have a closed form, then you resort to approximate inference approaches, which I'm not going to talk too much in this tutorial. Any questions so far? Great. Okay, so this is a graphical model of the Gaussian LDS, where X evolves in a Markovian way, which means xt only depends on xt minus 1, not the previous history. So we will have like this Markovian chain in the continuous latent states, and each condition on each of latent xt will generate yt using the Gaussian distribution, Gaussian emission model. Again, this looks exactly the same structure as hidden Markov model, but have continuous transitions and continuous emissions and everything like that. Which also means you can use the forward-backward message passing or the EM algorithm from hidden Markov model to solve this LDS. Okay, the thing I emphasize those Gaussians because there's really Poisson, right? We already see from the GLM, Gaussian is not perfect for spike trains. We need to have Poisson distribution. So very straightforwardly, replace Py given X by the Poisson distribution. This part is exactly the same as the GLM part. Okay, and there's another model called Poisson F. It's actually not linear dynamic system, but this paper proposed this method called PFLDS. Oh, it is a linear dynamic system, my bad. It's a Poisson F linear dynamic system. They put an F there. The F basically indicate the mapping from X to Y is nonlinear, right? You have a nonlinear function F on top of X. Okay, so this is the only extension. And to answer some question, F now is a learnable nonlinear function, can be parameterized by a neural network. So you want to learn the parameter of neural network to learn this nonlinearity. Okay, cool. So these are just three kind of LDS based. Oh, still is LDS because the latent dynamic evolves linearly, just the observation model is via a nonlinear neural network. So they have some linear, nonlinear mixture together. Question? Make sense? Okay, great. I just want to show some simple, interesting like data fitting of all these models. Here's one simulated data generated from a Gaussian LDS, right? You can generate a 2D latent X, the whole sequence here, right? It's a 2D continuous signal. And if you plot this X1, X2 on this 2D plane, that gives you this spiral shape. It starts from the top right and then spiral, swirl and converge into the middle. And this dynamics actually can be modeled or captured by a linear system. And then from the 2D latent, you can generate a 10-dimensional observation. Each one can correspond to a neuron, right? So if you do see a neuron like have a pattern like this, then it's likely there are only 2 different dimensions underlying this 10-dimensional neural population. Okay, here's a data fitting to the macaque V1 data. Now it's also V1, but we're not dealing with single neurons. We're dealing with the whole population of V1 neurons. Okay, so I still show the single neuron fitting because when you map from the latent to the neuron, you need to know the mapping for each neuron, right? For each neuron, how this latent map to me, right? So that's you still observe that mapping function, either in a linear way or in a nonlinear way. So you can actually plot the true firing rate if you have, which is the black curve, and then you can fit the model with the Poisson LDS or Poisson F LDS. Remember, the only difference is the mapping, the data generation is via a linear function or a nonlinear function using a neural network. You can see if you have a nonlinear generating function, usually it fits better, right? I mean, that's probably pretty intuitive. And on the right here is inferred latent in a 2D plot. So if you plot, assume the latent X is 2-dimensional, right? And then you can learn that X from this population of neurons. LDS give you some circle shape. Oh, the reason is actually because the stimulus is like a rotation, is a periodic like drifting stimuli. So the neurons are triggered in a periodic way. That's why you see these cycles. And if you look at the latent underlying these neurons, they actually give you these circles. And the color correspond to the face, I think. Color, oh sorry, the green color and the light color, like these correspond to different faces. And the stimulus like cycles many times, that's why you will see the switches between the green color, deep green and light green. Okay, but if you use a FLDS fit, you get cleaner latent trajectory, which probably because some nonlinearity is absorbed by the nonlinear observation function. And you can also plot this in a 3D space if you fit a 3-dimensional latent and got kind of interesting, like, I don't know, like cylinder shape of torus, or maybe torus shape of manifold. Okay, and of course, quantitatively, you can show the PFLDS fits the data better with higher likelihood, something like that. Okay, question? No? Cool, and they also show an application to this macaque reach out, center outreaching data, where the animal is actually in the motor cortex. The animal reach out to, I think here's seven different directions. On the left here is the behavioral trajectory of the monkey. Okay, they record the behavior trajectory, different color correspond to a different angle. And then they collect the motor cortex neuron, apply these two models to it, and find this 2-dimensional latent trajectory underlying the motor cortex. And they show that, especially with the FLDS, you see groups. Okay, each group might correspond to a certain reaching out direction, but use a PLDS, you might don't see that color separation very well. So sometimes people do want some nonlinearity in this kind of latent variable model. So although you can think of this as the underlying acts that trigger the neural activity, right? You don't observe this, you only observe the reaching direction. Well, if you fit the GLM, you can also use these the reaching trajectory as the acts, right, to fit the GLM model. But the cool thing here is I don't know acts even. I don't know what these neurons are doing. I have no clue about what could trigger these neural response. I just fit a model to it. Without any external information, I can find something like this. Okay, so this kind of cool because you can try to unbox these neural populations, which seems to be very irregular without any clear pattern, into something really, interpretable with nice manifold or shape. And this is good because when you do apply to some populations where you don't know that kind of external information, hopefully we can resort to this kind of tool to understand what these underlying neural dynamics. Okay, any question make sense? This part, question? Single trial. So the question about single trial average,
yes.
That's a really good question. So before you have these all fancy models, one thing you can do dimensionality reduction is using PCA, right? You can perform PCA, but that usually requires trial average. You need to average the trials, and then you will get a mapping and the low dimensional representation. All the models I'm talking about today are single trial. So you actually don't need to average the trials. You can just intake the single trial data, and you can learn this thing. So the good thing is you can now model the trial by trial variability or the trial by trial variance, which actually explain the data, help to explain the data. Okay.
Just to clarify, so each of the trajectory here is one neuron, a single neuron?
So this is the low-dimensional, two-dimensional representation of, say, 100 neurons behavior. You can think your original neuron is 100 by time. Now I map this 100 by time into a 2 by time matrix and visualize that 2 by time matrix. Each curve here is one single trial, which answered the previous question. Each curve is one single trial, and some trials reaching out to one direction, some trials reaching out to the other. That's why you have this group. And then when you want to recover, so the good thing about this kind of model is now I want to know for a single neuron, what this neuron is doing by corresponding to that specific reaching behavior. What you can do is you can put the tuning curve, which is actually learned from the encoding model, on top of the latent. Now you can know, given this specific latent, how this specific neuron responds. So you can know how each single neuron responds over the entire latent space. Some neuron may respond to a certain direction, some neuron might respond to all direction, but in a certain phase of the reaching task. You can all get that information by interpreting the encoding model of this latent variable model. Yeah, question? Any question? Question?
And in general, the manifold doesn't need to be two-dimensional, right? Like it's just, you can look at pictures of two-dimension, but so is there a way to interpret like higher dimensional structure? Do you like run a clustering algorithm and then try to?
Yeah, so exactly. If you do have a low dimensional, you can interpret, but in many cases, a 2D might not be the best data fitting, right? You might do cross-validation, select the hyperparameter, which is the dimensionality to be 10, for example. And then you still cannot visualize that for interpretation purpose, two ways. One is perform a PCA on top of it, because you might assume all this like sophisticated nonlinear mapping is already captured. Then all you left is in a pretty linear and Euclidean distance space. Then you can perform a PCA to learn something like visualizable, right, a 2D, or you can do clustering, as you mentioned, to map, like perform t-SNE or UMAP. But usually, yeah, t-SNE, UMAP is just some nonlinear way of doing dimensionality reduction compared with PCA. I mean, people want to show how powerful their model is, so they basically use PCA to show like, my dimensionality reduction already disentangle all this nonlinearity, so I can use a PCA to learn some to visualize something already, instead of resort to t-SNE or UMAP. 5 minutes, okay. Actually, yeah, let me quickly finish this part by introducing 2 more models, then we'll talk about the rest in the next lecture tomorrow. So here, okay, I finished about the LDS. I will introduce 2 more cases where we actually have nonlinear dynamic. Right? In the previous 2, we have a linear dynamic system, just changed the nonlinear mapping. But now, what if we have nonlinear dynamic? Here, I just want to motivate a little bit, like people study worm, right? See like worm have this kind of nonlinear-ish changing kind of behavior. And people also record their behavior and record the whole, like here is about 100 head neurons in the C. elegans. And people record their recording using calcium imaging. Okay, so there is some data like this. And then you can visualize the worm neural data. The y-axis is the neurons from the calcium imaging, axis is the time. And you show different neural recording for different worms. So this is one type of data people study. The interesting thing I want to introduce is, okay, so if you look at each neurons, you plot them, that give you the whole trajectory, right? The green, blue, yellow trajectory. And then you map them down to some low dimensional space. And then you can visualize that you can color different phase of the trajectory correspond to the certain behavior you observe. Because you observe their behavior, you can characterize that into 4, which is the ventral turn, reverse, forward, and dorsal turn, right? You observe that, so you can give the label of each phase, behavior phase, and you color them in this PC space of the neural data, and you actually see they actually kind of have some nice aggregation separation between different phases. So that gives you, and again, observe the entire neural trajectory, they definitely exhibit nothing like linear dynamic. So then people think like, if we can segregate the whole trajectory into multiple states, each state might follow a certain linear dynamic, but state switches with some transitions that might give you some nonlinear property. So that's what the switching LDS is. Switching LDS is literally put another layer of discrete latent states on top of the continuous. And discrete might indicate the 4 behaviors, right? That you probably know. But generally, when you fit the model, you don't know this. I just use that example as the motivating example. But here you have the discrete states, which can switches. Each state will correspond to a certain linear dynamic system. Here is a fitting to this neural, maybe I think this is a simulated area. So you assume the whole sequence switches between 1, 2, 3, 4, 1, 2, 3, 4, and given a certain states, it follows a certain linear dynamic model, a certain behavior of the C elegans, and generate observed neural activity from this continuous latent. Okay, now you achieve some nice nonlinearity among the latent trajectory. Make sense? That one makes sense? Okay, lastly, I want to talk about the last one, which basically uses a recurrent neural network to capture the nonlinear. You assume X evolves following an Rn, you can assume an initial distribution for the first state, and then all the following states follow deterministically of the previous. This is actually what is done by this Nature method paper, but you can, I think you can also totally just add a Gaussian noise or some noise to each step. But I think probably the RN itself capture a lot of nonlinear dynamic part. People don't add too much noise or stochasticity to it. So this is what people use a lot of called LFAS. The main part is using a recurrent neural network rather than LDS over the latent. But the observation model part is very similar, like a linear model as the previous ones. And they show applying to this 3-dimensional Lorentz dynamic, if you have y1, y2, y3, or the 3 latent dimensions, you generate observed spike from these 3-dimensional Lorentz dynamic. And you show that using PLDS and LFAS, the LFAS fits better. The main difference between LFAS and PLDS is they're latent. One evolves linearly, one evolves through a recurrent neural network. That's it. That's the summary of this part. We introduced the 3 linear dynamic system and 2 nonlinear one. The first nonlinear using switching state to achieve the piecewise linear. The second one just use a recurrent neural network to do that. I think we're right on time. In the beginning of the next lecture tomorrow, I will talk about another set of models based on the Gaussian process. Yeah, I'll just wrap up and finish today.
Thanks very much, Anqi. If we have any questions on Zoom, maybe we'll start with the Zoom attendees.
Could you elaborate more on how switching is different from continuous evolution in LDS?
Continuous evolution in LDS? Well, yeah, I think switching basically, I think switching basically means you have multiple LDS. So like the C elegans example, right? Each behavior, turning behavior, or like ventral turning and dorsal turning, they certainly follow different dynamics. So it doesn't make sense if you just have one continuous LDS to model the entire behavior, but it also probably over complex model if you have a very complex RN to capture the entire, because we sort of know each behavior itself is kind of linear, right? You can, because C elegans, the structure is simple, you can sort of use this linear dynamic system to capture its behavior. And I think that is a motivating example to show why people have switching linear dynamic system, which certainly capture more complex evolution of dynamics compared with one single LDS.
Thank you. One more question. How to interpret the meaning of the latent dimensions?
So that's a good question too. There's usually no meaning without interpretation, because the latent dimension is something we define arbitrarily. There's no physical meaning. And one thing people really try hard in this field is to interpret that. Can we assign some physical meaning to each dimension? That's actually similar to the disentanglement analysis in general deep learning. People fit autoencoder or VAE, like latent model, underlying a set of images and define 3 dimensions, but how these 3 dimensions correspond to the interpretable, like the meaning of the original image, maybe contrast, size of the object, the color and stuff. And we also try to get more interpretable and disentangle latent representation so that we can assign a meaning to each latent dimension.
So we'll break for today. Let's thank Anqi again for an excellent tutorial. We reconvene online and here tomorrow, 10 to 12.